Welcome to video 1 in our course B10 Python for AI. So in this video, we're going to go through the basics of this course in terms of resources, times and aims, as well as some basic concepts in terms of data management. Firstly, for this course, there are a large number of resources that are very, very useful. I refer you to the previous courses, one on tidy modeling with R. Although we're focusing on Python here, I think that's a good resource in terms of some of the concepts that are relevant here. We have the classic Folsey paper linked here as well as another paper on protein language models capturing the evolutionary potential of SARS-CoV-2. Here is a GitHub link of many machine learning models for protein, some different types of protein modeling. There's the AlphaFold 3 paper here with an illustrated example, a detailed example of it at this link. There's another link that's very useful in terms of discussing the details of transformers. And likewise on ESM2, there's also a link here that explains it. And some of these links obviously go way beyond the detail of this course, which is very much introductory. So what are we trying to do here? We're trying to learn about AI data and models in Python. We're going to consider how research questions feed into AI approaches and how we might try different models to better understand them. So basically what we're going to focus on is key areas of the data that we might use for AI models, SVMs, gradient boosting, NLPs, particularly focusing on protein language models, and then a little bit on deep learning. And what I'm going to highlight at the start is that this is very much an introductory approach to these areas where we're coming from background, having a more limited background in bioinformatics. So what we want to do is to write Python code to apply AI models and implement pretty standard models in AI models in Python and consider how if we try different models, we may get different outputs. And that kind of means that what we're trying to learn about this course is how to model data with using Python, using an AI is just a type of statistics in simple terms, and choosing a coding language for AI models such as Python, and learning how we can classify a set of bacterial data we're going to focus on in the practicals into two categories, resistant or susceptible. And by doing that, what we're learning about how we can apply models using support vector machines, gradient boosting, deep learning, and NLPs. So that's our kind of outline for how we're going to go through things. We're going to firstly talk about data in a general sense, and think that if we're trying a particular model, we need to justify why are we using that, what's the basis, and consider about the raw data that we're using. Do you have sufficient data to answer your research question? What approach you're taking in terms of the modeling? Why is that being used? If you tried a different modeling approach, would you get the same results or different ones? And to what extent is that true or not true? And then if you are able to make some sort of prediction or classification, is that relevant for others? And if you can think about if you're doing this in 5 years' time, would you be doing it differently? Um, all the questions feed into research integrity and considering what you're trying to do, why you're trying to do it, and what it has. So there are lots and lots of examples we can use for thinking about why we would want to apply AI methods. And one of these examples from the last time I gave was linking about sign language. Everyone who can sign can do it in a different way. And just like human language text or human speaking, um, there are patterns of ways of doing it that are often very general, but also very unique. There's a range of features, and that sheer number of features becomes quite complex, and that therefore naturally lends itself to tools that can grapple with such heterogeneous and large-scale data that has a central structure, but is varies as well. And there's also the classic examples of image detection for AI models, and that can be tricky at times to distinguish between one image and another image. And what we're also thinking about is how that could be applied to biological images as well, because that's the real aim of this course, where the data that we're looking at, though, is numerical data. So what we're thinking about also is about making our data machine readable, bearing in mind the steps that might be involved, that we collect our data, manage our data. And then irrespective of what study we're doing, we're going to have to do extensive cleaning and transform it in some way so that the scales of the different features are comparable to each other. We might do exploratory data analysis to verify that a data is of sufficient quality to analyze, and then do model selection after that to select appropriate models, optimize those models, refine that, and that's an iterative process where we might find that we've nuances we didn't expect at the final point. And then we want to communicate the results and visualize them effectively so that it has some sort of impact. An analogy we can give for this, which I've given previously, is cooking. So when we have data, we have a certain set of ingredients, and we may want to remove any ingredients that maybe are um, overripe or not good anymore. And that's often part of data cleaning. When we think about the appliances that we have, which are like the algorithms, they have a particular behavior, a particular procedure that they can apply to the data and cook them in the data in a particular way. And then we have recipes that we're following in terms of how we should use these algorithms, which are like our models. And we get different outputs. With any large scale data, you'll get different outputs. Even if things are very, very similar, random seeds at the start can make a big difference. And I think this analogy is very relevant here. We could have something like random forest models where the optimization can mean that we get slightly different results at the end. So this is an example of structured data that you should be familiar with. It also aligns with having a data dictionary to or data catalog to detail our data that is inside our data set. And these names are um, relatively consistent that we have the abbreviated name here on the left, like partial inflation. We have maybe the human meaning here in terms of partial inflation and it being clinical data. And this is the full explanation here on the right to explain it to a person who hasn't seen that before. So it indicates if a mouse showed up partial pancreatic inflation. So what are we thinking about AI-ready data, machine-readable data? Is that our columns, are the variables, our rows, are the observations? Typically, if we're dealing with structured data, our goal is to be accurate, not creative. One variable per column. If you need more columns, that's fine. Don't do calculations in your spreadsheets. That's your raw data. Your analysis data is separate. If you have multiple components within a single cell, like wild type underscore replicate 1, which is different from infected replicate 2 or whatever, you can create a new column and split them up and have the replicate identifier as one column and the wild type infected, etc. as another column. So what are our goals for making AI-ready data? To make a data dictionary, create a detailed data cleaning plan as to why you're cleaning it in a particular way, import the data set into a relevant coding language, visualize and explore it there to make sure that it's okay, and write code to do the manipulations. Don't do it manually. This brings us to the topic of synthetic data. So synthetic data generation is sometimes useful. It depends on the area that you're working in. In biology, it's rarely used, really. However, for AI models, you may have this issue known as class imbalance, where you have too few of the rare samples here in blue compared to the ones in green on our left. So that imbalancing means we can't apply particular models. Um, and our issue is that blue is rare and sparse, green is common and well defined. So one approach to do that, known as SMOTE, would be to infer more blue points. So this is known as synthetic minority oversampling technique, and it deals with continuous data and applies a linear model to that. So for example, if we have different points here in red, we get a random sample of our points from the rare group. We get the k nearest neighbors of each particular sample, so you might have k equals 5. If we choose a sample like x1 here, we can create a new sample s1, which lies along the line here between x1 and x2, and some random number that allows a bit of noise to be in there. So we're not just taking a simple linear function of that, we're allowing noise to come into that. And then we repeat this process until there's a sufficient number of samples for balance. And that level of balance doesn't mean that the numbers are equal, it just means that they're in a proportion that we can apply some statistical test. There's variations in that, such as borderline SMOTE, where it tries to fill in the points along the at the boundaries between the two classes. And it just focuses on the borders of the point because they're the ones that really matter in terms of differentiating the two groups. And there's Python libraries we can use to that. Scikit-learn and imbalance-learn can help us apply that. Another variation of this is called AdaSyn, and that's basically focusing on regions where there's fewer points, and it's trying to focus on regions near the common group. And its focus is really on samples that are difficult to identify to help delineate these two boundaries. And that's part of what we'll do later is kind of draw boundaries between two groups, visualizing two-dimensional space to see how that works. So issues with AI related data, we could do a whole course on AI ethics, where we're more focused on the statistical aspects, but it's worth bearing in mind, um, data provenance is very, very important. So, there's been at least two, clinical prediction models and stroke derivatives with no clear data origins. They're available in Kaggle and they've been used in 124 studies so far and one medical device patent. A group used, the paper link is below, used Tricode NLI to check this data, and there's a clear and problematic issue with lack of information data collection. The data is not realistic. For example, the stroke has 100,000 people, but only 8 different values for blood glucose, which is quite puzzling. And then when you visualize the data, as I mentioned before about the advantages of EDA, what you see when you plot the blood glucose levels, and across the patient identifier is they have a particular pattern. And you see at this point at somewhere around patient ID 36,000, the noise distribution changes, but it's a clear pattern. So looks like there's some sort of standard normal variable, putting data here, and then random data here. That's also here, and that's changed for the one down below. And it's clear that this does not look like random data. It looks very, very biased. And I just want to point out that we should be conscious of this issue. So, what we're going to do now is just quick recap briefly on supervised models in AI. So we have regression models that deal with continuous data, and you've heard some of them before: linear models, ridge regression models, lasso regression. There's different options. There's also nonlinear models, such as polynomial ones or other ones. And these are different from ones that models discrete data that is most associated with classification. So logistic regression is effectively the form of regression that we're using in this practical for the previous one in R and this one in Python. They can often use KNN, K nearest neighbors, decision trees, and random forest models to apply that classification. And then there's the whole area of tuning and model evaluation at the end. We talked about clustering before, and standard methods would be k-means clustering, hierarchical clustering, and dbScan. We also talked about dimension reduction, which would include methods like PCA, MDS, TSNE, UMAP, and to an extent NMF, which is non-negative matrix factorization, which is useful in biology because it works with positive samples that are non-zero. So, more things that we can recap on is ensemble learning. So that's think about things like random forest and boosting with XGBoost or AdaBoost. So this ensemble learning allows methods to be collated together to give some variation in the different models that could be applied. We'll also talk about support vector machines that allow, in the sense of that scattered plot before, us to classify two different data sets by creating a boundary between them. And that might use kernel methods to allow nonlinear effects to be present. I don't think in our last course we talked about graph neural networks, but they're a form of neural networks where they can manage complex biological data. A phylogenetic tree is a simple example of that. Here we might have, for a phylogeny, we might have nodes as the samples and ancestors, and edges denoting the evolutionary time or generational change. And the distances between these nodes is reflected in the graphs, and that allows us to use evolution relationships, transition dynamics, and reconstruct the trees as well. So you kind of see it here. We have a phylogenetic tree in the topology and a distance matrix here. And we could apply different node-based methods like logistic regression, random forest, and so on, or other methods that allow more sophisticated modeling. So more of our recap. So for Python, we will use Python notebooks in Google Colab. You can also use Jupyter notebooks. Key packages that are associated with that will be NumPy because this allows multi-dimensional arrays and it also allows linear algebra approaches to be applied. SciPy allows us to do statistical analysis. Matplotlib allows us to visualize the data effectively. Pandas allows us to do exploratory data analysis and manipulate the data. Scikit-learn uses NumPy and SciPy to do machine learning. So we'll use that. Um, yeah, so that's our short introduction to data for AI.